Hi everyone, today we'll be talking about the G proteins and the second messengers.
So, what are the G proteins? These are actually the middlemen between the first messenger and the second messenger. What they do is that they connect the receptor on the outside and the enzyme on the inside. So, why is it called the G protein? Why not the H protein or the F protein? This is because it is a guanine nucleotide binding that binds to the GDP or the GTP. So in the first messenger, we have the water-soluble hormone or our ligand acting as a cell surface receptor. Then it forms a ligand receptor complex. As you can see, the GDP is first inactive and is activated to GTP. Take note, our G protein is a heterotrimer. That means it consists of the alpha, beta, and gamma subunits. So once it's activated, our alpha subunit kicks out the beta and gamma subunit, and this activates various signaling pathways. But we're not going to go deep into that in this video. Now for the actual action, which are the second messengers. They act as amplifiers to take the signals from the outside and send to the nucleus. So our GPCR, that's our G protein coupled receptors
That activate the G proteins and simultaneously activating the adenylate cyclase. This converts ATP to cyclic AMP. Thus, our second messenger is activated. So the C-AMP can then bind to the protein kinase A. This kinase A has two regulatory domains and two catalytic domains. The catalytic domains are usually inactivated in a normal situation, just like our GDP. However, once the C-AMP binds to this regulatory domain, the catalytic domains are freed from this complex, then free to migrate into the nucleus. So what do they help with? Why are we learning them? They regulate the activation and the deactivation of the G proteins. In other words, they are responsible for the hydrolysis of GTP back to GDP. An important function of this C-AMP is for the formation of long-term memory. Do you wonder how scientists figured out that C-AMP could work out as a second messenger? L. Wilbur Sutherland, who received the Nobel Prize in 1971. A moment of appreciation for him, please. He worked on the hormonal mechanism, especially the epinephrine, by showing how nearby cells can secrete epinephrine, which will invoke signal inside the cell and then work with our C-AMP. So now a bit of history, guys. So when Sutherland moved to the Washington University with his collab, he discovered the epinephrine or adrenaline can lead to the breakdown of glycogen to glucose via the process called glycogenolysis, which happens in the liver. So he used a mouse liver, then he centrifuged them into two fractions, the cytoplasmic fraction and the membrane fraction. Obviously, the cytoplasmic fraction contained the enzymatic components and the membrane fraction contained the receptor molecules. Now, he added epinephrine in the membrane components, and we know that epinephrine results in some legendary second messengers. Then, he filtered out the solution and put it into the cytoplasmic fraction. It was discovered that after the addition of the epinephrine, it activated the membrane components in which the phosphorylase activity dramatically increased, telling us that there is a diffusible molecule in the cytoplasm which acts as a relay signal. So what could this relay molecule be? Pause the video for a while and think. Okay, so it turned out to be our C-AMP. Now we know when we have a need for rapid muscle movement, adrenaline is secreted. So it's our second messenger for today, calcium. We all know calcium is important for physiological processes. Ringa in 1880 was trying to compose several artificial buffer which can make an isolated heart stop beating. Isn't that interesting, guys? Unfortunately, his several experiments failed until one day when he used tap water to make the buffer instead of distilled water. He saw magically that the heart started beating again. Could you think why? It was discovered later on that the London tap water was heavily contaminated by the salts of calcium. Then Ringer's solution was made. It was then made to all the cellular aspects of calcium, like for muscle contraction, secretion, metabolism, fertilization, and many more. That's enough for the history, guys. So, for the cellular aspect, the GQ gets activated and activate the phospholipase C. That breaks down phospholipids in the membrane. A product of it is called the inositol trisphosphate. This migrates to the endoplasmic reticulum, and then our ER contains many IP3 receptors that bind to the IP3. And finally, the stored calcium in the AR are released to the cytosol. Congrats, guys, if you stayed till the end of this video. See you next time, and don't forget to hit the subscribe button. Thank you.